Good evening, everyone. I welcome you all to the week 12, which is the final class live session, live problem solving session on bioinformatics algorithms and application course. So, before moving, uh, uh, as we missed out in the last class, I'll be doing a Vika demonstration before, followed by the overview of the overall course. Yeah, to start with, uh, Vika is a popular open source data mining tool that provides a graphical user interface that provides a GUI. You're seeing a Veka graphical interface chooser to apply various machine learning algorithms to their datasets. So in the last class, we have discussed several machine learning algorithms. Some are classification based, some are regression based. So Veka is a graphical user interface, but which is a on-click or by-click option to do those machine learning tasks. So it was developed in University of Waikato in New Zealand and is available under general public license. So this Veka graphical user interface provides an easy to use interface for performing data analysis tasks without need for programming. So, so users can load datasets into the tool, pre-process the data, filter the data, and apply machine learning algorithms to develop the models or to analyze the data. Okay. And given here, the Weka also provides an explorer option, experimenter option, a knowledge flow option, workbench, and a command line interface. Like say for the users who prefer to work with the tool programmatically, they can use this simple CLI command line interface. So starting with the first Explorer tab. So the Weka Explorer is the graphical interface that allows users to perform various data mining tasks. Explorer contains several subtabs that provide access to different functionalities of the tool. So if you see the subtabs here, the first is the pre-process tab. This tab provides several options for pre-processing, data filtering, attribute selection. So attributes here are nothing but features that we use to develop the model, data normalization. Next is the classify tab. So this tab allows users to build and evaluate classification models on their dataset. Next, followed by this is cluster tab. So this tab allows users to perform cluster analysis on their dataset and to identify groups of similar data points. Users can choose from a variety of clustering algorithms such as k-means, hierarchical clustering, and visualize their clusters as well here. Followed by that, we have associate tab, select attributes tab, a visualize tab, and visualize tab. So overall, this Veka Explorer provides several options for data pre-processing. Provides a comprehensive set of tools and functionalities for data mining and machine learning analysis. Users can choose from a variety of algorithms and techniques to pre-process their data, build predictive models, and evaluate their performance. Next, to start with the classify tab. So as I mentioned before, this classifier provides a set of classification algorithms, or we shouldn't go by the name. This classifier or classify tab has the algorithms for the both regression as well as the classification problem. So how this has been selected? So classification problems has the nominal values, whereas the regression has the continuous values. So based on that, this classifier automatically filters out. So based on the data set that we give, this will automatically filter out what can be applicable. So in this we have classifier tab. So this window allows users to select an algorithm from a list of available options. And Veka does a background work for us, so it filter out classification and regression algorithms based on the data we input. So if you give a classification data set, this classifier shows only classification algorithm. If you give a regression data, it shows only regression algorithms. Next is the test set options. So for configuring the evaluation of classification or regression models. So here the model can be evaluated using the training set option, where the performance of the model is evaluated by the predictions made on the training set itself. The second option is the supply test set, where the performance of the model is tested on the new and unseen test set. Followed by that we have cross validation, and percentage split. Next, talking about the data. So we have instance and attributes. So number of rows or number of data we have termed as instance. Attributes are features. So as each software and each tool has its own file formats, Weka has ARF format, but still Weka takes the CSV format, comma separated file format. So under the pre-process option, you had an open file option. So from using this open file option, one can download the, one can load the dataset into the Weka environment. Once a data is loaded into pre-process tab, so the attributes, the instances, and the statistics regarding the data can be seen in the under the pre-process tab. So when you see, we have this instances window, so where you can see a number of instances, a number of attributes followed by the name of the each attribute is there. So using this remove option provided below the names of the attribute, one can select or remove the attributes. Then if you see on the right hand side, we have data statistics, where it is a number of district data, number of unit data, and what is the type of the variable that we selected, followed by that some plot related data. Next, several machine learning algorithms are there. So these algorithms are grouped based on their common functionalities. For example, Bayes function uses naive Bayes based probability. The concept of naive Bayes. When you take functions, these have functions such as linear regression, multiple linear regression, and other functions. So where we map the output variable as a function of input variables. Next, we have lazy algorithms. So lazy algorithms are nothing but as soon as you give the training set data, they won't train, and when they encounter the test data based on their training data, then they start learning. When they encounter the test data, they make predictions. A meta are the combined consensus models, which are combination of two or more. These are natural prediction trees, random forest, and other. And as I told, based on the dataset we input, this classifier tab automatically filters out the useful algorithms. Here you can see the MS rules has been marked out because it is not applicable on this dataset. If we take another dataset where MS rules has been applicable, we see 1R or part or some other algorithm is masked like this, like MS rule is masked here. And then different validation options that we had. So the widely used validation options are supply test set and cross validation. So cross validation is nothing but if you have 100 data and we use a 10-fold cross validation, 90% or 90% of the data is used for training and the remaining 10% is used for testing, and this process is continued until all the data once has been seen in the test set. So this process is repetitive process such that this 10% has in each repetition this 10% come across all the 100%, and in all these different iteration predictions the value is taken as average. This is this test set option. So the properties of the test set should be, it shouldn't be the part of the training set. So there is no meaning in giving the training set instances as test set instances because the model has been already encountered them. So it is very good model. So it is not an well way to judge our model. But the only care should be taken in the test set is that test set should have same features or same attributes as such of the training set. Otherwise, see when there is a no attribute or when there is a no value, the function cannot be built or the model cannot predict. Even how can we give a test set? So the test set also has similar format, but if you see, instead of predicting or giving the output variable, we give question mark. So if you give a question mark, it will predict. Like this is an unknown. But to further see, the more options can be used, and the way we need to judge the output, whether we need to know the statistics of the model, or we have to know the equation of the model, or we need to know the performance of the model. So based on different options are provided in classifier evaluation options. Yeah, so when you see here, this is the output. So here we have selected an SMO algorithm and the option of test set. So this test set is not part of training, and this is an alpha beta classification problem. And in the output tab, you see plus sign in error column, which shows mismatch. That means if you see plus means it is actually predicted as beta, but it's showing error plus. So the actual thing is this should be alpha. So here are some questions for you. So Weka is used for you can provide answers using the chat box. I'll be seeing that. Okay, 1 second. I think I have moved the slides. Yeah, the first option is Weka can be used for clustering, regression, as well as classification problems. Next, the output of Weka provides information on so Weka provides a sensitivity, specificity, and accuracy. So all of the information. So this sensitivity, specificity, accuracy are the evaluation metrics for a classification problem. So this is what the brief demo I would like to give about the Weka software. If you want a detailed explanation of the Weka and all, so we do even conduct NPTEL in collaboration with NPTEL, the workshop on Weka. It's a 2-day workshop where we train you and we provide you with some projects where students will get hands-on experience and hands-on learning experience, as well as hands-on working experience on Weka environment. So after this, I'll be moving on to the. So any doubts on Weka or any clarifications on Weka, I'll be happy to provide. Yeah, so starting with the concept of bioinformatics. So this is an overview. So bioinformatics is an interdisciplinary field that combines biology, computer science, statistics, and mathematics to analyze and interpret biological data. The goal of bioinformatics is to develop tools and algorithms that can extract meaningful information from large biological data sets such as DNA sequences, protein structures, and gene expression data. Nowadays, bioinformatics also plays a key role in personalized medicine, which seeks to tailor medical treatment or individual genetic makeup and many more. So, based on this, the bioinformatics has become an essential component of modern biology, providing the researchers with the tools they need to better understand the complex biological systems and diseases. So bioinformatics methods are used to analyze genomic and proteomic data to identify genes and proteins that are involved in disease process, to study the function and structure of genes, and to identify potential drugs. Some key features of bioinformatics include its interdisciplinary nature, that is, bioinformatics combined with multiple fields such as biology, computer science, and other things that we learn. Next, followed by that is big data analysis, as bioinformatics deals with the large data set of biological information, and because it has powerful computational tools and algorithms to process and analyze data. Next, advances in technology have been enabled the generation of large amounts of biological data at much faster rate and allowing high throughput analysis of biological systems. And followed by this in silico experimentation, data integration, applications in medicines, and rapidly evolution. So these are some of the key features of bioinformatics. So bioinformatics plays a crucial role in studying biological systems at different levels of complexity. From molecular to ecological, here are some examples. At the molecular level, bioinformatics is used to analyze and interpret biological data such as DNA, RNA, and protein sequences. Bioinformaticians use tools and algorithms to identify and annotate genes, predict protein structures and functions, and study the interaction between biomolecules. This information is critical for understanding the molecular basis of disease, drug discovery, and personalized medicine. At the cellular level, bioinformatics is used to analyze the gene expression data from different cell types and stages of development. So, by comparing the gene expression profiles, bioinformaticians can identify the genes that are active in specific cell types or at specific developmental stages and gain insights into cellular processes such as differentiation and proliferation. Next, at organism level, bioinformatics is used to study the genome and transcriptomes of different organisms. By comparing the genomes and transcriptomes of different species, bioinformatics can identify the genetic basis of traits and diseases, or study the evolutionary relationship between the species and develop phylogenetic trees. At the population level, bioinformatics is used to analyze the genetic variation within and between the populations. By studying the genetic diversity of populations, bioinformatician can identify the genetic basis of a disease and study migration patterns of populations. At the ecological level, bioinformatics is used to study the interactions between the organisms and their environment. Here, it helps to analyze the large ecological data sets such as biodiversity data, environmental monitoring data, climate data to understand the dynamics of ecosystem, predict the effects of environmental changes. So in computational terms, to talk about transcription. So it's a process of creating an RNA copy of DNA sequence. So this involves use of an enzyme called RNA polymerase to read the DNA sequence and synthesize an RNA molecule using complementary base pairing rules. Followed by that, we have translation. So this is a process of synthesizing a protein from mRNA molecule. And then we have a concept of a complementary strand of DNA. Followed by that, codon table. So this is a table that relates each codon in the mRNA molecule to a specific amino acid in the protein sequence. So in general, to tell about this, codon table is a set of rules that is used by the ribosome to synthesize the protein. That means the combination of these three nucleotides should form this amino acid. For example, AUG codes for amino acid methionine, while the codons UAA, UAG, and UGA are stop signals that signal end of a protein sequence. Then we started with the sequence-based parameters in the course. So there are several sequence-based parameters that is used to analyze a DNA or RNA complexes. The first one is the length of DNA or RNA, which refers to the number of nucleotides in the sequence. So sequence length is an important parameter for analyzing sequence data, as this can affect the accuracy and efficiency of computational algorithms. Followed by that, the GC content refers to the percentage of nucleotides that are either coherent or cytosine. GC content can have an effect on several biological processes, including DNA replication, transcription, and translation. Next is the codon usage bias, which refers to the preference of certain codons to be used more frequently than others to encode a particular amino acid. So codon usage bias can vary between different algorithms and can affect protein expression levels and protein function. The melting temperature of a sequence refers to the temperature at which a double-stranded DNA molecule dissociates into single strand. Secondary structure of an RNA refers to the way in which RNA molecule folds into three-dimensional shape due to base pairing interactions between nucleotides. The last one is sequence conservation, which refers to the degree of similarity between different DNA or RNA sequences. So conserved sequences are often indicative of functional and structural importance and can be used to identify functionally important elements such as regulatory regions and protein encoding genes. So these sequence-based parameters are critical for many bioinformatics analysis, including sequence alignment, genome annotation, and gene expression analysis. The next is databases. So databases are collection of data that are organized in a structured manner, making it easy to store, access, and retrieve information. In bioinformatics, databases play a critical role in storing and managing biological data, including DNA and protein sequences, gene expression data, and functional annotations. So generally the characteristics of a database include bioinformatics databases can store large amounts of data ranging from individual genes to proteins to entire genomes or transcriptomes. Next, data in bioinformatics databases are organized in structured manner using well-defined data models and schemas. So this allows efficient storage, retrieval, and analysis of the data. Bioinformatics databases are designed to be interoperable, meaning that they can communicate and exchange data with other databases and software tools. So this allows for data integration, analysis across multiple datasets and platforms. Accessibility. Many bioinformatics databases are publicly available, making them accessible to researchers and scientists worldwide. So this promotes open access to data and encourages collaboration and sharing of resources. Bioinformatics databases often integrate with analysis tools and software, allowing users to perform complex analysis and visualization of the data within the database environment. So the importance of bioinformatics databases cannot be overstated. They provide a centralized and standardized repository for biological data, enabling researchers to access, analyze, and compare data from multiple sources. This promotes a data-driven research, enabling the discovery of new biological insights and development of new therapies and treatments of human diseases. Moreover, the availability of these databases has transformed the field of bioinformatics, enabling researchers to tackle complex biological problems and accelerate scientific discovery. So talking about the nucleotide sequence and property databases, which store and manage information related to DNA and RNA sequences. So these databases play a critical role in analysis and interpretation of genomic, transcriptomic, and are essential resources for researchers working in the field of molecular biology. Talking about the nucleotide sequence databases, which contain information about nucleotide sequences of DNA and RNA molecules. So these databases include sequences from a wide range of organisms, from bacteria to humans, and provide a wealth of information about genetic variation, gene expression, and molecular function. Some of the most well-known nucleotide sequence databases include NCBI, EMBL, and DDBJ. Next is the property databases. On the other hand, These contain information about physical and chemical properties of nucleotides, such as their melting temperature, base composition, and secondary structure. These databases are useful for predicting the behavior of nucleic acid molecules under different conditions and can be used to design experiments or optimize protocols. So examples of these include RNA structure database, RNA secondary structure database, or oligocal database, many. We have a SIGTO feature as well, but that is regarding RNA. So in addition to these sequence and property databases, there are also many other types of bioinformatics databases that store and manage a wide variety of biological data, including protein sequences, gene expressions, functional annotations. So these databases are termed as critical resources for researchers working in the field of bioinformatics, providing a centralized repository of the data that can be used to answer important biological questions and drive scientific discovery. Just give me a second. Yeah, now starting with the topic of proteins. Proteins are micromolecules composed of amino acids that are folded into a specific three-dimensional structure. The structure of a protein is critical for its function as it determines how protein interacts with other molecules in the cell. So proteins can be characterized into several levels of structure, including primary, secondary, tertiary, and quaternary. So talking about the primary structure of a protein, which refers to the linear sequence of amino acids that make up the protein. The sequence of amino acids is encoded in the DNA of a gene that encodes protein. Changes in primary structure of a protein, such as mutations, can have profound effects on protein structure and function. Then we have secondary structure. Secondary structure of a protein refers to the way in which amino acid sequence folds into regular patterns, such as alpha helices or beta sheets. These structures are stabilized by hydrogen bonds between amino acid residues and can be predicted using bioinformatics tools. Next is the tertiary structure. Tertiary structure of a protein refers to the overall three-dimensional shape of a protein, which is determined by the interactions between the amino acid residues in the secondary structure. Tertiary structure is critical for protein's function as it determines how protein interact with other molecules in cell. Next is quaternary structure. The quaternary structure of a protein refers to the way in which multiple protein subunits come together to form functional protein complex. So examples of quaternary structure of proteins include hemoglobin, which is composed of 4 subunits, and other example is collagen, which is composed of 3 polypeptide chains. So this overall, the structure and organization of a protein are critical for their function, and understanding the structure of a protein is a fundamental part in the field of biochemistry and molecular biology. Similarly, we have some protein sequence databases. So, sorry, I think this is a repeat slide. Talking with the databases, UniProt and PDB are 2 important databases in bioinformatics that store and manage information about proteins. So talking about UniProt is a comprehensive protein sequence database that provides a centralized repository for information about protein sequences and functions. It is maintained collaboratively by EBI, Swiss Institute of Bioinformatics, and Protein Information Research. So this UniProt contains information about 200 million protein sequences from a wide range of organisms, including bacteria, viruses, and eukaryotes. One of the most useful features of UniProt is the ability to search for protein sequences based on specific criteria such as sequence similarity, domain structure, or functional annotation. And it provides an extensive annotation for each protein sequence, including information about protein structure, function, subcellular location, post-translational modifications, and also contains links to the external resources such as literature, genome databases, and many other things. Next is the PDB. contains information about three-dimensional structure of proteins and other macromolecules determined by X-ray crystallography, NMR spectroscopy, and other experimental methods. So overall, UniProt and PDB are two essential resources for researchers in the field of bioinformatics, and they provide centralized repository for information about the proteins, including sequence data, functional annotation, three-dimensional structure, and so this understanding about three-dimensional structure is important to answer biological questions and drive specific scientific discovery. Then talking about sequence alignments method, so which are used to compare and align two or more biological sequences to identify the regions of similarity and difference and dissimilarity. This sequence alignment can help in the identification of evolutionary relationships between the sequences. Prediction of protein structure and function is also done using this sequence alignment. So we'll see how this is done. Starting with the pairwise alignment, the first is the Needleman-Wunsch algorithm or the global algorithm. So this is the concept of dynamic programming. And then followed by we have a local alignment or Needleman-Smith-Waterman algorithm. And then basic local alignment search tool, BLAST. So BLAST is a widely used algorithm for pairwise sequence alignment that uses a heuristic approach to identify similar sequences. Then we have multiple sequence alignment. Also, multiple sequence alignment could be performed by many tools such as ClustalW, Muscle, tCoffee, and many other things. So we'll talk each of these methods in detail now, starting with the pairwise alignment methods. So the global alignment method are used when comparing sequences with similar length and conserved sequence domains. So these methods assume that the sequences being compared are evolved from common ancestor and may contain conserved regions. So this example is a Needleman-Wunsch algorithm, which is commonly used. So this algorithm uses a dynamic programming approach to determine the optimal global alignment of two protein sequences. It assigns a score to each alignment based on a scoring matrix that considers the similarity between amino acids. The algorithm generates a matrix of scores for all possible alignments between the two sequences and then identifies the alignment with the highest score. The conditions for global alignment are: the sequences being compared should have conserved regions, and sequences being compared should be of similar length, and also sequences compared should be evolutionarily related. So most of the times we use homologous sequences. Next is the local alignment, where contains we try to find the local regions that have or small regions of similarity. Smith-Waterman is a common algorithm that is used for local alignment. So and this is also similar, uses a dynamic programming approach to determine the local alignment. So conditions for local alignment are: sequences being compared may not have conserved regions and may be of different length and may not be evolutionary related. Followed by this, we'll be discussing about different mutation matrices. So PAM and BLOSSOM. So point acceptor mutations stands for PAM stands for point acceptor mutations. BLOSSOM stands for block substituted mutations. So these matrices provide a score of aligning two amino acid residues based on the similarity and frequency of occurrence in a set of homologous protein. So PAM is based on the assumption that rate of amino acid substitution is constant over time. So PAM matrices are constructed using statistical methods that analyzes the frequency of amino acid substitutions in a set of homologous protein. To construct a PAM matrix, the following steps are taken. A set of homologous protein sequences is aligned, and the percentage of times one amino acid is substituted by the another amino acid is calculated. So the probability of each amino acid substitution is assigned as a Poisson distribution. So the probabilities are converted to log odd scores, which is used to construct the PAM matrix. Next is the block substitution matrix or BLOSSOM matrix. So these are based on observation that amino acid substitutions are not constant over time, but rather depend on degree of evolutionary divergence between sequences. To construct a blossom matrix, a set of homologar protein sequences are clustered based on their sequence identity. For each cluster, AMSA multiple sequence alignment was performed, and the frequency of amino acid substitutions is calculated for each column of the alignment, that is for each position. So the probability of each amino acid substitution is estimated using a log-out score, and the threshold is used to define the boundary between conserved and variable positions in the alignment. So next we start with the BLAST and FASTA tools. So these are widely used algorithms for sequence similarity searches in mathematics. Both algorithms use heuristic approaches to quickly find the regions of similarity between two sequences and then use dynamic programming to optimize the alignment. So BLAST has these following steps. The query sequence is broken into small fragments called k-mers, and a precompiled database of sequence is searched for a matches to each k-mer using a fast algorithm based on hashing. Then sequences with a high scoring matches to k-mers are retrieved and aligned to the query sequence using dynamic programming. The highest scoring alignment are reported as hits. So this BLAST algorithm is designed for fast searching of large databases and is particularly useful for identifying homologous sequences in distantly related algorithms. Next is the FASTA algorithm. So here in the FASTA, the query sequence is broken into the smaller fragments called words, and again a precompiled database of sequence is searched for matches. So sequences with high scoring matches to the words are retrieved and aligned to the querying sequence using dynamic programming. So the FASTA algorithm is similar to BLAST, but it is more sensitive for detecting weak similarities between sequences. So overall, both BLAST and FASTA are powerful tools for sequence similarity searching, and the choice of algorithm depends on specific research question being addressed. Then we have phylogenetic tree. A phylogenetic tree is a branching diagram that represents the evolutionary relationship between a set of biological species or groups. It is used to study the evolutionary history of algorithm and to infer sequence of events that led to their diversification and speciation. There are several different methods for constructing phylogenetic trees, each with its own strength and weakness. So it's the maximum parsimony method. So this method seeks to construct a tree that requires fewest number of evolutionary changes to explain the observed sequence data. It is assumed that the true tree is one that requires the least amount of change from a common ancestor. Next is maximum likelihood. This method seeks to construct a tree that maximizes the probability of observing the sequence data given a particular tree topology and evolutionary model. It assumes that the true tree is the one that is more likely to have generated the observed sequence data. Next is the neighbor joining method. So this method constructs a tree by iteratively joining the pairs of taxa that have shortest distance between them. It assumes that the true tree can be constructed by repeatedly joining the closest pairs of taxa. Followed by that, we have a distance-based methods and a Bayesian inference as well. So each of these methods has its own strength and weakness, and the choice of the method depends on specific research question being addressed. The characteristics of the sequence data and availability of computational resources are also affects the method we select. In practice, it is common to use multiple methods to construct and compare the decision trees in order to identify the most robust and well supported tree topology. So protein sequence analysis include sequence alignment, phylogenetic analysis, secondary structure prediction, tertiary structure prediction, and motif and domain analysis. Till now, we have read about sequence alignment and phylogenetic analysis. Hereafter, I'll be discussing about secondary structure prediction, tertiary structure prediction as well. Before that, we learn some concepts. So the first concept that we are going to learn about hydrophobicity profile. So hydrophobicity profiles are the plots that show hydrophobic and hydrophilic properties of protein along its length. So these profiles are created by calculating the hydrophobicity of each amino acid in the protein sequence and plotting it as a function of position in the sequence. Hydrophobicity profiles are important for understanding the three-dimensional structure of a protein and function of proteins, as well as for predicting the localization of proteins within the cell. Next is protein patterns. So protein patterns are short amino acid interruption. Are the short pattern of amino acid sequences that are conserved across the multiple proteins with a particular family or a group. So these patterns are important for identifying the function of uncharacterized proteins based on the similarity to new proteins, as well as they are useful for predicting the evolutionary relationships between different protein families. Next is an PSSM matrix, which is position specific scoring matrix. Is a table that summarizes the frequency of occurrence of each amino acid at each position in a protein family or group. PSSMs are created by aligning multiple sequence alignments with a family or a group, and then calculated the frequency of occurrence of each amino acid at each position in the alignment. So PSSMs are important for identifying conserved regions and motifs within the proteins, as well as for predicting the function of an uncharacterized protein. Then that we have profiles which are similar to PSSMs in that they summarize the frequency of occurrence of each amino acid at each position in the protein family or a group. So profiles are more sophisticated than PSSMs as they take into account the likelihood of amino acid substitutions at each position based on evolutionary history. Profiles are important for identifying the conserved domains and motifs within proteins, as well as for predicting the function of uncharacterized proteins based on their similarity to known proteins. So hydrophobicity profiles, protein patterns, PSSMs, and profiles are all important tools for analyzing protein sequences and predicting their function. Next, we start with the concept of introduction to redundant and non-redundant sequences. Redundant sequences refers to multiple copies of same sequence that exist in a sequence database. This can happen due to errors in sequencing, annotation, or deposition of sequences. Redundancy can complicate data analysis and slow down database searches. So it is important to remove redundant sequences from databases whenever possible. Non-redundant sequences, on the other hand, are unique sequences that are present in a database. These sequences are useful for a variety of applications such as sequence alignment, gene prediction, and functional annotation. So construction of a non-redundant, and of course we are not constructing from redundant sequences, we are constructing and getting a data set of non-redundant sequences. So there are several steps. First is a database of sequences created by collecting all the available sequence from a particular species, gene family, or other group of interest. So this dataset is then cleaned by removing low quality sequences or sequence with errors or inconsistencies or duplicates. So once the database has been cleaned, a clustering algorithm is used to group similar sequences together. The clustering algorithm uses a threshold value of sequence similarity to group and that are likely to be homologous. Sequences that are above the threshold value are clustered together, while the sequences that are below the threshold value are left unclustered. So CD-HIT is the widely used tool for clustering and removing non-redundant sequences from a sequence database. It uses a fast algorithm to cluster the sequences based on user-defined threshold of whatever the user can give. I think the base cutoff is from 30 to 99. So CD hit creates a representative sequence for each cluster, which is usually the longest sequence in the cluster. All the other sequences in the cluster are considered redundant and are removed from the database. So CD hit can be used to create non-redundant sequence databases that are useful for a wide range of applications such as sequence alignment, phylogenetic analysis, and functional annotation. So the tool is fast, accurate, and easy to use, making it a popular choice for many bioinformatics applications. And then we start with the methods of secondary structure prediction. So there are several techniques used for protein structure secondary structure predictions, including Chow-Fasman methods, which is based on the observation that certain amino acid residues are likely to be found in certain secondary structures than others. So this method attains a probability score to each residue in the protein sequence for each secondary structure element based on their frequency of in known protein structures. So the scores are then used to predict the secondary structure elements in protein sequence. Next is the neural networks. So these are machine learning based algorithms that can be trained on large data set of known protein structures to predict the secondary structure element. So these algorithms use a variety of features including amino acid composition, sequence conservation, physiochemical properties of amino acids just to predict the secondary structure. So SVM is another machine learning. So it's and hidden macro model, again they are probability based models. And at last we have some hybrid methods which are combination of individual techniques such as neural networks or SVMs or HMM or SVM. So these combinations have been done to improve the accuracy of secondary structure prediction. Next is the protein. Just give me a second. I'll come. For the German class. So now we are starting with the protein structure prediction. This is a challenging task in computational biology as the number of possible conformations of a protein can be enormous. However, several computation methods have been developed to predict protein structures ranging from low resolution methods to high resolution methods. So starting with the homology modeling. This method is also known as comparative modeling and involves predicting the structure of a target protein by comparing its amino acid sequence to the known structure of homologous protein. Homology modeling relies on the assumption that proteins with similar sequences have similar structures, and it can be an effective way to predict structures for proteins that have close homologs with known structures. Next is the ab initio modeling. So this method involves the predicting the structure of a protein from first principles without relying on any prior knowledge of its structure. Ab initio modeling uses ab initio modeling uses various algorithms to search for the lowest energy conformation of a protein based on its sequence and physiochemical properties. This method is computationally expensive and can be less accurate than homology modeling for larger proteins. Next is the fold recognition. This method involves the predicting the structure of a protein by identifying its fold or overall shape based on its amino acid sequence. Fold recognition relies on the fact that certain proteins folds are more common than others and that proteins with similar folds often have similar functions. Fold recognition algorithm compares the target of a protein sequence to a database of known protein folds and select the best match. Next we have molecular dynamic simulations and hybrid methods. Hybrid methods are nothing but combination of about two or more. So overall, predicting the protein structure remains a challenging problem in computational biology, but advances in algorithms, computational power, experimental techniques are making it increasingly feasible. So then we'll be talking about various tools and techniques used for analyzing the protein tertiary structure. So the first is the visual inspection. So this involves visually inspecting the protein structures using software such as PyMol and Chimera. This can help and identify important structural features such as active sites, ligand binding sites, and protein-protein interactions. Next is electrostatic surface calculations. So this involves calculating the electrostatic potential of the surface using Poisson-Boltzmann equation. This can help to identify regions of a protein that are positively or negatively charged and which can be important for ligand binding or protein-protein interactions. Then we have solvent accessible surface area calculations. So this involves calculating the surface area of the protein that is accessible to solvent molecules. So this can help to identify the regions of a protein that are buried, exposed to the solvent, so which can be important for protein-protein interactions or ligand binding. Next is protein-protein interaction predictor. So this involves predicting the interaction between two or more proteins based on their three-dimensional structures. So this can be done using software tools such as docking, which uses algorithms to predict the optimal orientation of two protein structures in order to maximize. So additionally, again, molecular dynamic simulations and Fibonacci calculations are nowadays techniques that are used to analyze the protein structures. Then we have protein stability. So protein stability refers to the ability of a protein to maintain its folded conformation under different environmental conditions, such as change in temperature, pH, and ionic strength. The stability of a protein is determined by the balance between the energy required to maintain its folded conformation and the energy released when it undergoes conformational changes. Protein stability can be affected by the mutations in the protein sequence, which can disrupt the interactions that stabilize the folded conformation. The effects of mutation on protein stability can be classified into two categories. Destabilizing mutations. So these mutations reduce the stability of the protein and increase the likelihood of it unfolding. So destabilizing mutations can result from changes in amino acid sequence that disrupt hydrogen bonds, hydrophobic interactions, and electrostatic interaction. Stabilizing mutations. These mutations increase the stability of the protein and reduce the likelihood of it unfolding. Stabilizing mutations can result from changes in amino acid sequence that strengthen the interaction that contribute to protein folding. So the effects of mutation on protein stability can be quantified using experimental and computational methods. So experimental methods include thermal denaturation assay, circular dichroism spectroscopy, and differential scanning calorimetry. So these experimental methods measure the changes in protein stability under different environmental conditions. So computational methods include free energy calculations or machine learning methods or molecular dynamic simulations as well. So understanding the effects of mutation on protein stability is important for many applications, including protein engineering, drug design, and disease diagnosis. By predicting the effects of mutation on protein stability, researchers can identify mutations that can be used to improve the stability and function of a proteins or diagnose genetic diseases caused by mutations. Finally, I would like to discuss about CAD. So it's a computational approach that uses algorithms and simulations to design and optimize small molecules or drugs for specific disease targets. The goal of CAD is to identify drug candidates that have high affinity selectivity for their target protein as well as good pharmacokinetic properties. So CAD involves the first step in CAD is to identify the protein target for a specific disease. This can be done to various methods such as genetic analysis, proteomics, and literature searches. Once target of a protein has been identified, its structure is determined or modeled using X-ray crystallography, NMR, or homology modeling. This step is crucial for understanding the binding state and interactions between the target and potential drug molecules. In this step of virtual screening, library of small molecular compounds is screened against the target protein using computational methods. Virtual screening can be done using a variety of techniques such as docking, molecular dynamic simulations, and machine learning algorithms. Next is the hit identification and optimization. After virtual screening, the compounds that bind most strongly to the target protein are identified as hits. So these hits are then optimized through chemical modifications to improve their potency, selectivity, and pharmacokinetic properties. So this step involves a combination of experimental and computational methods. Next is the ADMET property check or ADMET prediction. So ADMET stands for absorption, distribution, metabolism, excretion, and toxicity. So this is the final step in CAD, which is to predict the ADMET properties of the optimized drug candidate. So this includes predicting how the drug will be absorbed, distributed, metabolized, and excreted by the body, as well as its potential toxicity. The CAD has several advantages over traditional drug discovery methods, including reduced cost and time, increased success rates, and ability to design drugs with higher affinity and selectivity of their target proteins. However, CAD also has some limitations, such as the need for accurate protein structures and the challenge of predicting ADMET properties with high accuracy. So, despite these limitations, CAD is an increasingly important tool in drug discovery and has led to the development of several successful drugs. So, here have some instructions for docking. So, docking is a method used to predict the binding orientation and affinity of small molecule to a target protein. So, the goal of docking is to identify the optimal orientation of the ligand that maximizes the interaction energy between the ligand and the protein. So, docking typically involves a preparation of the target protein and the ligand. The protein and the ligand structures are typically prepared by uh, removing the water molecules and adding missing atoms, bonds, or charges. Then a library of possible ligand conformations is generated and protein is scanned for its potential binding sites. Then in all these possible ligand conformations, these ligand conformations are scored based on the interaction energy between the ligand and protein. Then all the possible conformations are ranked based on their interaction energies, and top scoring conformations are selected for further analysis. So docking is a useful tool in predicting the binding orientation and affinity of a single ligand to a target protein, but it does not consider the flexibility of protein or the ligand. Next is the virtual screening. Virtual screening is a method used to identify potential lead compounds for drug discovery by screening a large database of small molecules against a target protein. So virtual screening includes preparation of a target protein. So that is removing water molecules and adding missing atoms, bonds, and charges. Then is a generation of ligand library. A database of small molecules is generated, which typically includes millions of compounds. Then is search for potential ligands. The ligand library is screened against the protein using various scoring functions. Then ranking of ligands. The ligands are ranked based on their predicted binding affinity to the protein. In summary, docking and virtual screening are two computational methods used in drug discovery to predict the binding affinity of small molecules to a target protein. Docking predicts the binding orientation and affinity of single ligand to a target protein, while virtual screening identifies the potential lead compounds by screening a large database of small molecules against the target protein. Next, we have pharmacophore modeling and QSAR, which nothing but quantitative structure activity relationships, which are these commonly used computational methods in drug discovery and design. Pharmacophore modeling is a process of identifying and characterizing the structural and chemical features in a ligand that are necessary for its biological activity. The goal of pharmacophore modeling is to develop an hypothesis about the active site of a target protein and to use its information to design new ligands with an increased potency and selectivity. The pharmacophore model is usually generated by analyzing the three-dimensional structures of non-active ligands and identifying the common features that are important for activity. So these features may include hydrogen bond donors, acceptors, aromatic rings, hydrophobic features, and many more. So once pharmacophore model is generated, it can be used to screen virtual compound libraries to identify new lead compounds. QSAR, on the other hand, is a statistical modeling technique that reveals chemical structure of a compound to its biological activity. The goal of QSAR is to develop the quantitative model that can predict the biological activity of a compound based on its chemical structure. Okay, so with this, I'm done with my explanations. So if you have any questions, please feel free to post in the chat box or unmute yourself and you can ask. Before that, I would like to thank each one of you for making this problem solving session a grand success, and being patient to the sessions and attending the sessions. This is the last session for this time. Yeah. Now I would like to take some questions. You can put it in the chat or you can unmute yourself. Yeah, if no other questions, the people are free to leave. Before leaving, all the very best for your exam. Yeah, you can ask questions from any topic, not even from this or any other doubt you have regarding this NPTEL. Okay. Wait for some more time. Okay, should I expect no other questions? Every one of you are very well prepared for the exam. Okay, I'll be closing the call now then. If anybody are typing, do let me know. Okay, yeah. So I have a question here. So where the question is about calculation of radius of gyration. So when you take a radius of gyration, it's generally mostly this the studies of radius of gyration. These are done in molecular dynamics simulations. But how come a radius of gyration can be calculated for a given static state of a protein? So that is how our understanding will differ. And the way the radius of gyration defined in what I mean there in simulations is very different. Just give me a second. I'm searching for a slide to show the formula. Yeah. So generally, when you take an radius, sorry, I couldn't search for the formula, but I hope you can get the formula in PDB param of our tool. So this is a tool developed by our lab. So the name of the tool is PDB. So PDB PARAM, PDB param, and you can see the documentation provided over there. So it gives us the formula. So it is generally start. Yeah, so it is generally given as a mass into difference in its coordinate position along with the center of mass. So center of mass is nothing but, so we have a formula for calculating center of mass as well. So based on that, you calculate center of mass first, and we have xyz coordinates, and center of mass also will have xyz values, and we have mass of the atom or the amino acid we are calculating. So based on that, we calculate the radius of gyration. So radius of gyration is square root of sum of the product of mass into difference of its coordinate with the center of mass divided by the sum of the masses. This is how radius of gyration is calculated. And this is a single value, but in simulations you see it continuously calculates for each time-based configuration confirmation it gets. Please do refer PDB param for the detailed formula and explanations. Then I have, yeah, of course, I'm not doing the assignments will affect, but don't worry, you can do well in the exam and cover it up. Yeah, even I do. This is me second time teaching a live session, and but first time experiencing a continuous audience. So even I see till date some of my first class people still here. So a special thanks to them who have been continuously attending. Yeah, whatever you see, yeah, center of mass, radius of gyration. Yeah, it also has XYZ for each. So here I have a question. So mentioning that like center of mass, radius of gyration. So when we calculate x center of mass, x coordinate of center of mass, y and z. So similarly, it has to be applicable. Actually, there's a wonderful paper published by Ghosh in 2009. I hope this is the paper should be cited in the PDB param as well. So where they have developed this wonderful concept of radius of gyration. So for detailed analysis, I think that paper would be helpful. A 2009 paper from Ghosh. If you have no other questions, you guys are free to leave. And if you think typing is taking a lot of time for the questions, please unmute yourself and ask